Hello everybody. It's my great pleasure to be here today to share with you the work that was done in my group over the past couple of years to develop multidimensional LC workflow for the next generation of process analytical technology for biotherapeutic products. But before diving into this technology with you, I wanted to go back on what's a process analytical technology, also called PAT. For the FDA, it's a system for designing, analyzing, and controlling manufacturing through timely measurement of critical quality and performance attributes of pro and in-process materials and processes, with the goal of ensuring final product quality. For that, we'll have to develop online and automated process with integrated continuous bioprocess that will allow us to decrease the preparation time and increase the process knowledge in order for us to improve the final drug quality. In that system, the quality is monitored through the process rather than tested at the end. This vision is a joint vision for multiple industry leaders. The goal is to implement the infrastructure that can allow to have a complete batch disposition package. For that, we will have to move from a static approach to a dynamic approach. In that sense, we will increase the process control and robustness, but also identify issues before the impact batch release. Where are we with this overall technology? What is the current state, and what are the current challenges with regard to PAT for biologics? We know that most PAT are focused on improving the means of cell culture process performances and parameters, but very few can directly measure the product quality attribute, such as oxidation, deamidation, or glycosylation. PAT technologies have been mainly focused on tools in the form of sensor or spectroscopic probes, and we know that these spectroscopic techniques often require the use of predictive chemometric models, often product-specific, and typically require large data sets to be validated and need to be re-optimized and or revalidated when the manufacturing process conditions change. In addition to that, as already mentioned, with standard manufacturing process, product quality is only tested at the end, and the standard lead time for batch release is about 30 days. So why are there just very few analytical tools that can measure the protein CQA? We know that MABs have much higher diversity and complexity compared to small molecules. And there's not a single analytical platform able to identify or monitor all the secrets simultaneously. Yes, LC is the main analytical workhorse for protein therapeutic, but the application of chromatographic methods that can simultaneously monitor multiple secrets have been lacking mainly due to time-consuming and offline sample preparation. So combining automated sample preparation with peptide mapping method can solve this identified bottleneck. That's where multidimensional LC can come to help us by combining automated sample preparation with multi-level analysis on the same system. For that, we can implement in the first dimension a huge diversity of chromatographic modes, such as ion exchange, SEC, IC, or affinity chromatography. After that, a reduction level is performed as well as a digestion on trypsin or lysyl colon. And at the end, the LP colon is used to do peptide level separation before MS detection. It's also important to know that we can directly link the first and second dimension to the MS in order for us to get intact and reduced level information by to the LC-MS. The system is fully automable and there is no manual external intervention for the sample preparation. And it's important to notice that only one technician is required for running this analytical system. Before diving into the application of this workflow for online monitoring, I would like to point out that we have been adopting this type of approach, combining sample prep with multi-level analysis across our company Roche Genentech, and we have reached a global alignment on how this workflow can be used to perform extended characterization of minor variants such as charge variant. In that case, we implemented in the first dimension an ion exchange chromatography colon followed by UV detection in order for us to identify and collect the peak of interest in different loops of our system. After that, one by one, the loops were injecting in the rest of our system in 2D, 3, and 4D to do reduction, trypsin digestion, and RPLC separation before MS detection. Like that, we were able to obtain different chromatogram and different MS information associated directly to the different fractions collected after the first dimension. Like that, we were able to correlate peptide level with protein level information. On this study, we compare the results generated in our lab in US with other labs in Germany or Switzerland. And on this slide, we can see how the same workflow, which is the peptide mapping analysis of charge variants, could be achieved even using different vendors and configuration. A high sequence coverage is obtained across all labs between 95 and 97%. The results obtained in the 4 labs were in agreement between each other and with offline data. So this data confirms the potential of this workflow for fast and reliable PTM analysis. Following this result, we assess the potential of multidimensional LC-MS for the analysis of cell culture samples. For this proof of concept study, 10 ml samples were drawn from the bioreactor of 2 liters over 14 days. As I mentioned, this is a proof of concept study in the sense that there is no physical connection between the bioreactor and the multi-DLC system. The samples were collected and prepared offline. Concerning the multidimensional LC-MS workflow, a purification step by protein A was first assessed before reduction and digestion on 2 different colon, trypsin or lysi, and RPLC final separation for peptide mapping analysis before MS detection. On this slide, we can get a good idea of the quality of the data generated with this workflow. On the left, we have the overlay of the protein A data with 12 injections, showing how highly reproducible the system is. Then in the middle, we can see the sequence coverage for trypsin on top and lysi at the bottom. The missed cleavages were about 2% for lysi and slightly higher for trypsin with 7%. Lastly, on the right, we can see the TIC profiles of the tryptic and lysi digestion. Unmodified peptide selected for the assessment of the multi-DLCMS method repeatability are denoted by asterisks. Overall, the data were highly reliable and could be used for PTM analysis. To test that the overall system is working as expected, we analyzed the standard MAB and various PTM were measured by peptide mapping, such as termination or succinate formation. Here, we are reporting the data obtained for the day 10, 12, and 14, and each sample was analyzed in triplicate and compared to the reference data, analyzed with the standard offline peptide mapping method. For example, the oxidation on the heavy chain was relatively low and constant, indicating that the process was robust and the product was under well-controlled consistent oxidative stress. We also see that we were able to monitor change over time, such as VHS and terminal lysine that were decreasing over the 14 days, but also the deviation that was slightly increasing with the time. The take-home message is that there were no significant difference compared to the offline method. The ASD were below 8%, and more importantly, this study is showing that we can use such system to measure PTM of cell culture samples. The ability of multi-dimensional LC-MS to monitor changes in the glycan profile for the MAB was then evaluated relative to the standard offline procedure, ELIC fluorescence. A total of 9 N glycans were identified by both approaches at similar levels. Overall, good correlation between the two assays were obtained, but low-level glycans of less than 0.4% were only detected with the ELIC assay. However, N-glycan analysis by multidimensional LC show really satisfactory results. All the data we have seen until now were obtained on a standard IgG1, but this workflow can also be applied to more complex formats. Here, for example, we have an Fc fusion protein with 4 N-glycan linked sites per cytokine. On the right, we can see these sites a little bit more in detail. And also, we are showing why it's important to use two enzymes, lysine and trypsin. Indeed, asparagine one site is close to the tryptic cleavage site. And to avoid loss of information caused by serine hindrance due to N-glycosylation, multi-enzymatic digestion were performed. To characterize PTM, a first series of experiments was performed using the immobilized trypsin cartridge, since it's the most widely used protease for bottom-up proteomics. The levels of peptide modifications, including oxidation, deamidation, proline amidation, and succimide formation, were measured. Similarly to the previously analyzed IgG1, various PTM were measured across the production days, analyzed in triplicate, and reported here. The results show a good correlation between the offline and online data for the analysis of the reference data, with RSD values lower than 7% for the online approach. And finally, PTM levels could be successfully measured over the production process. The key feature of this assay is that we can evaluate the site-specific information and describe the distinct population of glycan variants at the site. In this case, we were able to evaluate antinority, co-fucosylation, and sialylation of the Fc fusion protein. A total of 29, 28, and 27 N-glycans were detected for the asparagine 1, 2, and 3 respectively. For example, degree of branching was different between the sites. So we can say that multidimensional LC-MS was able to provide site-specific information which will be lost upon releasing and pulling the N-linked glycans by traditional offline methods. To go further with this technology, we decided to combine the sample prep with multi-level analysis capacity of this multi-dimensional LC system directly to the bioreactor to finally get a truly online system. For the connection with the bioreactor, we used a Numera system that was drawing samples from the bioreactor at specific intervals. Samples were filtered through a membrane to remove cells and then stored in an integrated liquid envelope in vials. Samples were then injected onto the multidimensional LC, and we were able, because of the facility of the fully automated and online multidimensional workflow, to gather information at multi-level analysis of maps directly from several in-parallel bioreactors. Fast characterization of several quality attributes within the same LC system was possible. For example, a titer measurement was done in 10 minutes, and characterization of several PTM, including charge variant at the intake level or glycation or glycosylation at the reduced level, were performed in 70 minutes. Finally, characterization of several attributes such as deamidation or oxidation at the peptide level were done in about 100 minutes. Here on this slide, we can see the various assessment criteria that we took into consideration to select the Numera. We were looking at the contamination rate, the occupation cell density upper limit, the clogging and carryover in flow path, the sample generation time, and the ease of use. And we finally get to work with the Numera because we eliminate the difference in sec flow and mass by the fact that they were prone to clogging or with difficult hardware and high maintenance needed. The performances of the multi-DLC connected to the bioreactor were evaluated on multiple criteria and on multiple samples coming from different days of culture duration. A high and consistent sequence coverage was obtained. However, a slight increase of miscleavage over time was observed, but that slight increase did not impact the mass accuracy. Indeed, a high mass accuracy of the MS instrument and consistency between multiple samples were obtained. To finish, online monitoring of cellulation for the Fc fusion protein was performed. A decrease in higher order cellulite species for the control of the cultivation day was observed and correlated with offline expectation. Furthermore, the addition of zinc is known to increase the total sialic acid concentration, and that's what we observed in our system. The process conditions can easily be compared to determine the impact of inputs on the quality of outputs. And finally, our online system is able to offer the novel capability to make in-process decisions upon determination of the sialic acid concentration. To conclude, we can divide what we can achieve with a multi-DLC system in three major parts. First of all, a fully automated workflow is achieved through connectivity to the bioreactor and by the capability of performing sample preparation and analysis. Then versatility with multiple digestion enzymes and chromatographic mode can be used on the same setup, allowing multi-level characterization. And finally, multiple PTM could be measured to evaluate trends over time. Before ending this presentation, I wanted to share with you how we can further enable the implementation of multi-DL CMS for PAD. And first of all, by use of simple detector. Indeed, the use of high-resolution MS should be mainly dedicated for method development purpose, and we should rely on more simple detector for testing, like QDM. Also, we should partner with vendor to implement software solution more tailored to PTM monitoring. And finally, we should use multi-D LCMS to complement spectroscopic-based measurements in the bioreactor to enhance the accuracy of the chemometric predictive models. When we are looking toward the future, we should continue to advance the inline and online technologies to achieve the full potential of adaptive process control, robust manufacturing, and real-time release testing. This should allow us to reduce batch release time by using a fully automated and integrated manufacturing and testing system. Like that, we'll be able to spread out production across several facilities rather than producing large batches in a single facility. To end this presentation, I wanted to acknowledge all my partners from Roche, Genentech, but also the University of Geneva.